I started the company in a time when startups weren't, hot or cool yet, at least not in biotech, and I didn't fit the profile of a biotech CEO. I could have been a founder, I guess, but someone who would be sort of behind the scenes, and then not really in control of the company. But I started Perlara as the world's first, biotech PBC, and it's been an incredible journey since. But at that time, I was really um, looking for something to actually work on. I didn't study rare diseases in my training at all.
Hi, Ethan. Thank you for being here.
Yes, my pleasure. Sorry, I just missed you by a few days, I know, but we'll be back in Vitalia soon.
Yeah, we are already waiting for you to come back. So, my first question is how it happened that you became a scientist? Because I know that you have also a PhD in philosophy, I think. Yeah, is it philosophy?
No, no, I did
His,
undergrad, history and
uh-huh.
sociology. Yeah.
History and sociology. Yes. So you have a PhD and undergrad in history and sociology, and then switched to biology.
Well, I
So
never really switched is the thing. I always wanted to be a scientist, and I used like the college years to broaden my horizons, and especially on the, quote unquote humanities or social sciences. And in the summer between semesters in college, I worked in a lab, and I told myself that's how I was actually training to be a scientist. So I had every intention to do a PhD in the sciences. I just wanted to enjoy my college years intellectually and broaden my horizons.
I see. So you wanted to be a scientist since you were like a kid.
Yeah, no, definitely. Yeah, I have memories, and my parents would confirm that I wanted to be a scientist from a young age. I had a chemistry kit. I was into marine biology for a while, which is why I
Wow.
got into scuba diving from a young age. So yeah, I was always into the sciences.
Interesting, because I'm always fascinated how uh, the story of creation of a company is connected to a personal story of a founder. So it's usually like that. And
Yes.
and then in 2014, you decided to leave the academia. So you were already a postdoc, and you left the academia and started a company to search for cures for rare diseases. How did you come to this decision? Because it was not common these days.
Uh, I I I decided that I didn't want to keep trying to get an academic job and just break in somewhere at some university and work my way up the, through the system, so to speak, to, to the kinds of research institutions I had worked in myself and had gotten accustomed to and had trained in. So I sort of closed that door and then opened up this door as a founder. But I didn't realize that I was actually a founder throughout the whole time that I was a scientist and being trained. I just didn't understand that there were other ways to be a scientist than as a professor at a research institution. So I broadened my personal horizons and started a company at a time when there was no such thing as founder-led bio. There was no real encouragement from my research institutions, from undergrad to grad to postdoc. There was no cultivation of entrepreneurship at that time. It wasn't something that was really discussed. You either got an academic job or you kind of quietly disappeared and nobody talked about you. And you probably end up having a great life in policy or science writing or countless other things that people do with PhDs. But only a small percentage of PhDs actually get tenure track positions. Um, so I started the company in a time when startups weren't, hot or cool yet, at least not in biotech, and I didn't fit the profile of a biotech CEO. I could have been a founder, I guess, but someone who would be sort of behind the scenes, and then not really in control of the company. But I started Perlara as the world's first, biotech PBC, and it's been an incredible journey since. But at that time, I was really looking for something to actually work on. I didn't study rare diseases in my training at all, and didn't think of myself as a rare disease scientist. And the real genesis of it all was meeting a mom whose twin daughters at the time had a very pernicious and ended up being fatal rare disease. And I was having a conversation with her at the NIH during Rare Disease Day, which is the one day of the year at the end of February where we're supposed to all remember that there are this plague of rare diseases. And I met this mother and started chatting about her disease because I happened to actually know something about the gene involved in the pathway that was affected. And after a few minutes, I thought, am I talking to a mom of two kids with the disease, or am I talking to a fellow scientist? Because I couldn't tell the difference if I closed my eyes, and realized, wait a minute, if this is a general feature for the most part of every parent whose child is struck by genetic lightning, then it dawned on me that I wanted to build a business, and I wanted to focus science on this problem because I would partner with these amazing people. And at the time, and even really still to this day, I don't think it's an insight that the rest of the world has really caught on to yet. It's not just me, and I wasn't probably the first, for sure. There were others before me in biotech, like Henry Tremere, who definitely understood the rare disease problem from the community, family, patient level. So definitely I pay homage to people like that. But yeah, the way that I wanted to sort of set it up as a PBC and to do all these other more radical experiments on the company side, that seemed like, that had to happen after I met this mother, because then I met a few others and realized it wasn't just a fluke. This is a generalizable phenomenon, and I wanted to build a business around partnering with these people.
Yeah, I think this is that sounds so logical, because I can imagine that every parent who has a kid with a rare disease, obviously he or she tries to read everything that is possible, to know more, to find out if there is a cure or any hope. So obviously they are forced to become do-it-yourself scientists.
Yep.
And to understand the nature of disease, at least to have a chance for their kid to survive.
Correct.
And then you developed this idea that you can test the potential cures in simple organisms, so yeast, worms, and flies. And
Yes.
this is a brilliant idea.
It's like a very small Noah's Ark, just a Noah's Ark for the small animals, for the tiny creatures.
Yeah, but usually mice are the ones who are suffering for all the science.
That's right. So we can make yeast suffer. We in fact, how do we know they don't enjoy this process? We use yeast to make bread and wine and to enrich our lives. So I see this more as a symbiosis and a partnership. And definitely it's better on the soul to pour a tube of yeast down the drain versus having to kill a mouse. So the yeast and the worms and the flies. So the reason that came about is I told you I wasn't really trained in rare diseases, but I did do my thesis work and my postdoctoral work with yeast and human cells as well. And was open to testing in other systems like worms and flies, although I didn't have direct experience. So I brought this experience with yeast, and no one was really using yeast as disease models in quite the way that I wanted to do at scale. People had thought about it on a gene by gene basis, and other people had thought to use yeast as disease models, definitely not claiming to be the first there at all. But in the case of trying to take a gene that was really not studied at all and use a yeast model to very rapidly generate drug repurposing candidates that families would be eager and willing to test, that's certainly not something that academics were doing. And it occurred to me that was going to be, if Perlara was going to have some kind of edge, or if its platform was going to be unique, it would be around this insight. And again, I wasn't the first to have this general insight about model organisms. It's a company called Exelixis, which still exists today, but it started in the late 90s by a bunch of geneticists, actually. And they had similar insight that worms and flies, whose genomes had just been sequenced in the late 90s, were going to be this great resource for disease modeling, not just basic biology modeling, but disease modeling as well. And that was the thesis of that company. They were ahead of their time in so many different ways. But again, Perlara was not really inventing any one thing per se de novo, but it was combining them all together in a unique way, in a unique combination. So with yeast, and then I added worms and flies and human cells to the platform over time, although we actually started with an emphasis on worms and flies to begin with. And with that set of that portfolio of disease models, we started to go after groups of diseases, in particular these metabolic diseases, which I know we'll talk more about. And then it also, it wasn't lost on us that these metabolic diseases involve very ancient proteins, very ancient pathways. All cells, all animal life does certain metabolic processes almost exactly the same. So we felt we were on solid ground scientifically using yeast and worms and flies to model these human metabolic diseases in children, because the underlying biology was very ancient and was preserved or conserved in something even as distantly related to humans as yeast. And we last shared a common ancestor 1 billion years ago. So there's been a lot of cousins in between. And in spite of that, our metabolisms in many ways are the parts are interchangeable. It's actually quite remarkable. So that really was the genesis of the company and the idea that's really been powering a lot of our efforts for 10 plus years now.
This is amazing. So this similarity is the reason why you actually can use the potential cure candidates in humans, and it can be efficient.
Correct. Exactly.
That's super interesting. So, and then you came up with an idea of this collaborative science where scientists, clinicians, and families, they all work together to find treatments for rare diseases. So this is what is now called Maggie's Pearl.
Yep.
Tell me a little bit about who is Maggie and about her family. I think that they're very special.
Yeah, we met this family, the Carmichael family, through a connection. Dr. Matt Might, who had a son, Bertrand, who passed away from not the same disease Maggie has, but a sister disease, if you will. And that connection was made in 2016. Maggie's mom, Holly, and Matt actually met in the White House back then, at the end of the Obama administration, the end of the Precision Medicine Initiative, which was a program that was coordinated or run out of the White House to raise awareness about precision medicine, and in particular how it could help rare diseases, really rare diseases. So that was the original connection, and I think we entered into a research agreement in early 2017, and everything was centered around Maggie. And she has PMM2. It's a form of glycosylation disorder, sugar processing, sugar metabolism disorder. And she, we can talk more about her specific case, but, her family was very motivated to do research. And we started working together on yeast and worm and fly avatars. Patient avatars is another way we describe them. And we started that work in 2017. And over the course of 17 and 18, we made a discovery of a molecule that was never approved in the United States, but was approved in Japan for a different purpose, in an entirely different patient population, adults actually. But it's not a random disease. It's actually adults suffering from diabetes, which is actually a chronic metabolic disease. And that was sort of the first hint that these pediatric metabolic diseases and these common chronic metabolic diseases could be biologically linked. So we started to think about how to take the results from the lab into the real world with Maggie. And over the course of 2019, we put together what's called a single patient IND, a single patient study. And this wouldn't be possible without Dr. Ava Morava-Kodic, who is our collaborator and clinician scientist, who is the doctor, who is administering this treatment and hopefully others like it that we will find. And maybe I'll just pause there because I could keep going on, but that's sort of how we created Maggie's Pearl and what happened to create it.
So you used so-called N of 1 trials. Explain a little bit, like for dummies, for me, what is that and why it's unique?
So I guess the idea of an N of 1 study is not really that new at all if you go back. I mean, I guess if Leonardo da Vinci were doing experiments on himself or somebody, those are maybe some of the first recorded N of 1s. And there's been Nobel Prizes that have been awarded as a result of N of 1s. People don't necessarily know all this history, but Australian doctor, I believe, Barry Marshall, won the Nobel Prize for proving that ulcers are caused by a bacterium because it hadn't been known. And the way to really prove it was to essentially drink a flask that contains the bacteria that causes the ulcer versus a control flask. And I think he was the first to sort of cast a stone, if you will, and that was an N of 1 in its truest form. So and in fact, there's N of 1s happening all around us. Maybe we don't see it. Every professional athlete is pursuing a kind of N of 1 to extend their professional career. They have doctors, we call it sports medicine, but they have very precision, personalized diets and exercise routines and other experimental interventions, including things like stem cells and peptides and other biologics. So they're running N of 1s. Maybe we know the N of 1 messiah, Brian Johnson. He's doing probably the most famous N of 1 in existence today, a study on himself and treatments or interventions he's using to treat his own aging. So this idea is out there. And in the rare disease world, it really coalesced around a mom named Julia Vitarello and her daughter Mila. There was a medicine made just for Mila called Milacin. This was in 2016. And it was right around that time when I see this example of an N of 1 in the rare disease world where it was literally one patient. So N of 1 made sense, but it wasn't immediately obvious, at least to everyone at the time, to me, that they were going to generalize that and say, well, even if there is a disease that doesn't happen to just affect one person, if there is a disease that affects multiple people, but you could start with one person and then scale from there. That was, I think, my radical shift in thinking, and that was really around that time, 2017, and that's when Perlara and all of our efforts started to focus around let's find the best possible test case, and the family and the science and everything had to work out to find a kiddo who we would say let's try an N of 1, let's do this ourselves, and so that's really, I think, the N of 1s in a nutshell. But I would just say I try to move away from that language, actually, and I flip it around, and I would say 1 to N is the better description of what we're doing. Because N of 1 gives you this sense of finality, singularity, that's just, there's one purpose. How could you possibly extend that to anything beyond? Instead of having that mental model, I want to have this expansion model where you start with 1 because you're comparing 1 to 0. You're comparing a vacuum to a point in space. And yes, you can't draw a single line through that point in space yet, But let's imagine you get a second point in space, a second N of one. All of a sudden you can draw a line. And then if you get a third and a fourth and so on, you can get a more precise fit. But I like to sort of talk about 1 to N, because that really says it doesn't really matter what N is. Maybe N's only going to be 5 in this disease. But in this other disease, it could be, all humans, because it's aging. So the 1 to N model is elegant, I think, as it fits everything from very small numbers to very large numbers.
I think it's also very inclusive, because if it's 1 to N, so it means that you can start with only one person. So you don't need like a huge trial. Yeah. So you are enough, or like one patient who is suffering from a specific disease is enough. Yeah. But why is developing a new drug so expensive? I heard different numbers. Yeah, like over 1 billion, over 500 millions. So why is it so expensive? And I mean, like, why it takes so long to create a new drug?
Yeah, I think this is there's a lot of factors that are at work, but at the end of the day, the way that drugs are discovered is sort of a Frankenstein process. And, the discovery part is happening usually in one place, like in academia. The preclinical work is usually happening in another place, like a small startup. And then the clinical work is happening at a bigger pharma company. And that alone, having to move between those institutions, creating business models at each of those steps, all the different scientific expertise you have to do to run the process end to end. There's a lot of reasons why it can be expensive, or I should say that it is expensive. But it is expensive because it can be expensive, but it can also be cheaper. And I think that's the flip side of what we're showing. But, for the most part, the current system is just very inefficient, especially at capital allocation and usage. A lot of companies, a lot of new VC funded startups, so to speak, will actually raise tens of millions of dollars because they have, they hire expensive C-suite, executives who are going to make hundreds of thousands of dollars a year in compensation, who, by the way, are constantly being, groomed for other positions, which they can happily take at any time. So just to even start in science, we're sort of shown that it costs tens and tens of millions of dollars just to ante up. Only certain people allowed to play this game. Not everyone is invited to this club. And then, of course, there was the cost of the earlier research that was done. That's happening in academia. It's publicly funded. It's happening over potentially longer timescales because academics aren't motivated by getting drugs approved. They're motivated by other factors. So you can just see the inefficiencies piling up, and the capital inefficiency in particular. So that's why those numbers are so high, is just the starting costs, the entry costs, the maintenance costs. Biotech companies might be burning millions of dollars a quarter just to operate. And when you add it all up, of course, the rare disease opportunities, these ultra rare disease opportunities, make no financial sense. They're economic suicide to pursue. And so if you're a rational shareholder, and so that's why you see companies, very big pharma companies, gravitating now toward very large blockbuster drugs like Ozempic and GLP-1 agonists, and looking at big markets like, no pun intended, like obesity and metabolic disease in adults. So they're going to happily feast on those large markets, and even if they tried to take on these rare diseases, they're just too big. They're too big to scale down to the opportunity, which obviously the solution makes itself apparent now, because if you have these rare disease families, these ultra rare disease families, they start with zero. They know they start with zero, and they build just what they need, and they move as fast as humanly possible. Essentially, they're the opposite of everything that pharma is today, which is why I think the cost structure that we're used to hearing, $1 billion, decade, these timescales shrink down when you have individual parents at the helm. Julia Villarreal, this example with Mila and Millicent, I mean, they developed this medicine for one child, but they did it, in essentially the span of a year. So that tells you that, and this was years ago. This was almost 8 years ago now. So that tells you that was what the technology allowed then. And so it really proves that it's not really about, the science or our gaps in knowledge that are holding us back. No. It's just someone who can move lightning fast and spend only what they need and focus on one, because as you said, one is enough. And if it actually is enough for one, it might be enough for others.
And I would say that it perfectly fits into the definition of a startup. So spend only what you need, build as quickly as possible, and be ready to pivot.
Exactly
And if
right.
you build for one, yeah, it can be used by others.
Hey, it's a small break for an announcement. We at Marabou decided to unite forces with Ethan Perlstein and his company Perlara. Our goal is to fund his research to find cures for 2 rare genetic diseases and help kids affected by these diseases. Our official campaign starts on 1st of September, but the pre-campaign is already open. We invite you to participate. You can do that with as little as $50, but all the help here is invaluable. Check the link in the video and description to know more and come to fund this important research. Thank you.
So, and here, drug repurposing comes into play, what you are doing essentially. So, can any drug be repurposed? I mean, can you discover new, I would say, applications for every existing drug, or it's something very special?
I guess I'm a maximalist. I think every drug will have a second or third or nth purpose, and some drugs will be outliers, and they will have many purposes. And maybe that's exactly
drugs.
maybe that's exactly what we're looking for. For example, in a longevity drug, something like rapamycin, which might impact overall aging. If it's going to affect overall aging, it means you're going to get all these other diseases fixed just coming along for the ride. And so there will be these, you can already look at statins, which are used in multiple diseases, not just necessarily high cholesterol, but in the context of heart disease or other conditions. So there are already examples of these outlier medicines, these medicines that have this crossover potential. And I'm a maximalist, so it may actually turn out that there are a handful of drugs out there that are so custom made, especially if you're talking about certain classes of programmable medicines. But I would say for the most part, it will be the exception rather than the rule that a molecule really just has one purpose. And I think we'll especially find that drugs that were developed a long time ago, or drugs that were inspired by nature, because they were synthetic versions of natural products. I think those in particular will be shown to have what's called polypharmacology. They'll have sort of multiple identities where they do multiple things. They have multiple effects on different targets, on different pathways, and maybe the dosing and the timing of the dosing, the frequency of the dosing will all matter. But with drug repurposing, I'm sort of a maximalist and think, at least, of every drug that we have today, and one certainly coming down the pipeline, they will have just every drug, small molecule drug at least, will have this intrinsic repurposability, just sort of a property, right? Because if it's just a pill, and all you need to do is move it from mouth to mouth, it doesn't matter. You can test many diseases. So I think there are others who agree with me, and I think there's folks like Evercure that are taking an AI centered approach, where they're going to make predictions for every drug, or maybe say for every disease, make a prediction for drugs, a list of drugs, because there's never going to be a magic bullet. It's if anything is, whether it's AI or even if it's based on a yeast model, it's still sort of a guess. It could be hallucination, right? It has to be confirmed in the real world. It has to be sourced, fact-checked, triple-checked. But yeah, I'm a maximalist. I think there will be multiple uses of every drug, quote unquote, and then drugs that are super bespoke and super personalized, we'll be able to just manufacture those at scale. That they'll practically be, they'll have those same benefits of repurposability, where you can just essentially spin one up at very low cost, and you can do that for any use case.
So, can you name me like few examples of drug repurposing that you discovered yourself, except Perlstein?
Yeah, so there's a few that we've talked about on our Substack. I think for some of these, I can't go into all the specific details quite yet, but for a disease called PIGW, which is related to PMM2 in a way, we found a particular vitamin and a particular nutraceutical, a combination of two over-the-counter, GRAS compounds, generally recognized as safe molecules, that we have this N of 1 result so far that you can look on our Substack and you can see video evidence of sort of before and after. I think that's one we're planning to hopefully actually in a few weeks add a few more families to the study. So going from 1 to 4 in this case. So that's an exciting one. There's another disease called PIGN, where we found a different vitamin in combination with an old generic, actually, that has a completely different purpose, and that combination, at least, well, for one of the two agents in the combination, I just got word today that there might be up to 8 families that have been sort of trying this over-the-counter nutraceutical, and there's a few other cases. There was a case that we talked about on Substack as well for ibuprofen, where we found for another CDG, another glycosylation disorder, the over-the-counter pain medication ibuprofen, an N of 3 study was run showing that
Wow.
there were treatment effects. Although that in that case, not all of these journeys are night and day. Sometimes we have to figure out the right dosing, the right frequency, and it's not always the dosing and the frequency for the original use. But with ibuprofen, we have that case study. For another disease, the folic acid was sort of the main event. So yeah, I think at least from yeast, almost a dozen times, I think we've gone from a yeast screen result to a N of 1 study, and where we've seen either some positive effect, or we've seen multiple families try and report some consistent effects.
That's crazy, I would say. Who would have thought that, as an example, folic acid has a potential to cure or significantly improve a condition?
Well, I would caution. I'm very comfortable with using the word cure, but I think the way I think about drug repurposing is, we should probably set the expectations as these will be treatments, and that maybe we get lucky and there'll be cases where if we could treat a baby, or treat very early, that maybe you can start to use stronger language. But I would say for now, we like to think about repurposing as a treatment option, as like a base hit, as a way to build up the foundation, raise the baseline, make the body and mind stronger to receive the next generation therapy, or the next wave, the second wave of therapies that we think will be, more targeted, having a higher expected effect, and maybe things that we ultimately would want to one day call cures. But yeah, just wanting to clarify, because not afraid to use the word cures. I just want to explain how we get there in a way that's actually doable.
Yeah, of course. But I believe that even the slightest improvement, an extra months, years of life for every parent of kid with a rare disease are super valuable. So how many compounds do you usually need to test to find a promising candidate?
Yeah, we've done this over a dozen times now, and we've done it also with worms and flies. At least with yeast, we look at around 8,000 molecules When we've looked at human cells, we've looked at around 6,000. When we've done this with worms, it's a few thousand. And with flies, it's around 2,000. So the more is better always, always, always. But in each of those cases, we found something. And I think there's maybe only one instance I'm aware of, and that's just of all throughout my entire network. There's one instance I'm aware of where, and this is maybe one out of, depends on the denominator, let's say 1 out of 35, maybe even 1 out of 40, there was nothing that came out of the screen. So it's rare, maybe, only a few percent probability of happening, but it could happen. But for 95+% of the time, we find something. And usually we find multiple candidates, and we have to have an additional triage process. But ultimately, certainly with just the yeast cases we've done, 12 or so yeast cases, in every case we found something. And half the time, it's an over-the-counter nutraceutical. So something, no further laboratory experiments are required, actually. You can go straight to N of 1 in those instances.
Yeah. So in case of Maggie, you definitely had a success. So what was the before and after with Maggie?
So if you look on my Twitter, my X, I have a video, a montage, mostly of her movement walking, and that's where we saw, gross motor improvement. There were other. If you look at a publication that was done about her N of 1, I believe it's in the Annals of Neurology from maybe September 2021, let's say, or somewhere around that time. There's maybe an example of her drawing a spiral and tracing and coloring. So there's plenty of kind of real world examples. And there was an ataxia assessment that was done, and she improved on that. So we have hard numbers to sort of back up what we're seeing with videos and drawings and so forth. And there's laboratory findings as well, from the blood, from the urine. There are other indications that we're having treatment effect that are based on hard, measurable outcomes.
Yeah, that's great results. So how did you and everybody around felt when you saw the after?
Yeah, I mean, we immediately wanted to figure out a way to have other kids get access to this, and then we realized the way to really do that is to run a phase 3 clinical study, which is ongoing and what we're doing now. And hopefully, we have still some more time in the trial, but hopefully we'll have our data readout and our submission to FDA, and we will present the case we have. And I think we are encouraged that you can start a trial, a phase 3 trial, based on N of 1 data effectively. So it sets a precedent, and but running a phase 3 trial is very different than running N of 1s. There's you have strict protocols, and you have clinical site visits. There's travel and other burden involved on the participants. So it's a big ask, right? And we're dealing with different ages of kids, different severity of disease, different mutations. But we kind of accepted all comers, and I think this represents the reality of this entire metabolic disease landscape, where you're going to have, maybe you're only going to be able to recruit a few dozen families into a study. So you want to be sure that you're moving forward when you have good justification. But we also have to figure out, how do we run more of these trials in parallel for other diseases? PMM2 is first because it has the largest number of patients, but there's other diseases like PIGN that have a large number of patients too, relatively speaking. We want to move on that front. So we don't have doctors just lying around, right? That's also a bottleneck. How do you start to find the right physicians? And they have to build relationships with the families as well over time. So, I think we're getting really good at N of 1s and N of fews, and we're starting to really parallelize that process. But the next the next stage in evolution is, now how do we start to do multiple phase 3 studies, let's say, and what's that network look like? How are we funding that? And of course, I've got plenty of ideas for that, but for now, we're sort of scaling the one to n as much as we can.
Yeah, and somewhere in the journey of Maggie's Pearl, so there were you, the parents, and the Mayo Clinic. So how did they come in?
So everything came together because the original sort of partners were Perlara and Maggie's family. They created a limited liability company that was sort of the joint partner, and then we brought in Mayo Clinic because that's where Dr. Eva Morava Kozic, our clinician, Maggie's doctor, actually, her and one of the world's leading experts in this area of diseases. Her institution at the time was Mayo Clinic. So the Maggie's single patient study was coordinated, and like the IRB and all of the approvals, that was all done through Mayo institutionally. So that kind of explains how they joined our partnership, and that's where the phase 3 study, which is currently ongoing and is now actually going to have a second site at Mount Sinai, which is where Dr. Morava is now has changed institutions, but at the time Mayo was the academic medical center partner, because that's where our clinical trial, which was then a single site, was located.
Yeah, so for Maggie, that was a life-saving story. And what diseases are you going to cure next?
So again, I think I would say definitely life-changing. We also know that sort of Maggie doesn't have the most severe form of the disease, thank goodness. But I just want to have the sort of proportional sort of statement here to what's happening. But yes, I think definitely life-changing, and we're still actually working on second and third generation treatment that we think will be much more cure-like or curative. But we definitely want to bring these life-changing and potentially, in some cases, life-saving treatment options to families sort of as responsibly and quickly as we can. So we're excited to, look for other types of partners who could help us, for example, with funding some of these projects, because right now we rely on families and their small communities to support the research. But the reality is not all families and communities can find the resources to fund this kind of research, even though we try to do it essentially at bare cost and to do it as fast as we can. It's still technical, specialized research, and there's still a cost to it. But I think, we're at the stage now where there's lots of interest in what we're doing. I think the more examples we can show, going not just from single patients, but to multiple patients, and recording the data, sharing the results, I think there's a lot of excitement for figuring out a way to do this at scale. Because in Maggie's disease, PMM2, there's 199 other sort of sister diseases that are in the same metabolic pathway. For mitochondrial diseases, there's several hundred genes as well that are involved in mitochondrial maintenance, biogenesis, function. Those all are very well modeled in yeast, for example. So I think we're at that threshold of scaling.
Yeah, that's exciting. I know that you plan to, but you would like to decentralize this process, and combine Perlara with Web3. Can you tell me a little bit about that?
Yeah, I think the Perlara has survived this long by evolving and pivot, I guess, is the technical term, but it's also, we've in some ways just pivoted in a massive circle, because it's still the same idea, but just changing the business model, changing the operating model, changing these other variables to figure out, and then just the timing has to be right. So I've always been interested in Web3 and interested in the potential for these very small communities to essentially, have governance rights and ownership rights as these distributed collectives, because every rare disease community is by definition distributed and decentralized. People live in different places and they have different backgrounds and expertise. So they really have all the makings of a great distributed team. If you see the benefit of their situation. So for Web3, just like with any technology, just like with the internet itself, there have been sort of boom cycles, some busts. There's been growth, maturation. There's been a lot of wilding, a lot of speculation, and potentially, not even potential. There has been, fraud in broadly in not necessarily Web3, but let's say crypto. So I've been watching the space, but now I think the time is right, and the technology is still both mature enough and in the long scheme still nascent enough that this is actually a great moment to do serious building. So I think Perlara is looking for ways, multiple ways, I think, to sort of lay down roots in a Web3 soil. I'm not going to do everything at once, but for example, on the funding side, it would be good to figure out ways where we can tap communities, crypto communities, and be able to finance some of these projects, so we don't have to rely on individual families to do this sort of backbreaking fundraising, or get lucky that there's a family with means that can just kind of self-finance it for a while. But even at some point they run out of money. So I think, thinking about crowd investing, a crypto angle, a blockchain angle, I think we definitely want to run an experiment there, or multiple experiments. Sort of on the NFT side, I think, patents have already been put on chain. Patents are what create value for investors in biotech companies for the most part. So I think running an experiment where we can put patents on chain is something we want to do as well. And I think there's other startups in the space that are exploring other ways to apply blockchain around biomedical assets, cell lines, even data. So I definitely want to do some experiments there. And then I think probably the last experiments we'll run will be sort of based on, we'll have to do with sort of governance, I should say, and tokenomics. I think those are still the parts of the system that need the most maturing, but at least when it comes to, I think, certain kinds of sort of crowd investing and for sort of digitizing biomedical assets, I think those are ready for prime time, and I want to explore those.
Yeah, that all looks like an ocean of opportunities for Perlara to thrive. I know that you work also with NARS, and ARS type of diseases. And when you were here, you said that they are somehow connected to longevity. How
Yes.
did you find out?
Yeah, there was a podcast by Dr. Matt Caberline. It's kind of, I think, one of the premier scientists in the longevity neuroscience field. Happens also have kind of trained classically as a yeast person. So we both share that sort of biological grounding. Also, his lab works with worms and has a company now that does worm longevity screens. As a springboard. Well, yeah, obviously as a springboard for doing studies in higher animals, of course. But I think that's obviously a great idea. And so he has a podcast, and recently was discussing these molecules called tRNA synthetases, which are, broadly speaking, part of metabolism. If you think about creating a protein, that's a metabolic process, of course. And these tRNA synthetases, they are these essential molecules that a yeast cell produces, a human cell produces, and every animal in between, plants for that matter, and any living thing that has to convert DNA into protein in terms of a translation process that they call translation, that requires these tRNA synthetases. You can think of them as like letters. So imagine your Sesame Street lesson in the alphabet. To make a protein, the language called protein, that has 20 letters. The language called DNA has 4 letters. Language called RNA has 4 letters too. They're basically the same as the DNA letters, except one difference. But you have to translate the from speaking DNA to speaking protein. And these tRNA synthetases are like these little translators, and they physically sort of link the two codes, the DNA, the information stored in the DNA code, and the information that is sort of manifested in the protein sequence. So these tRNA synthetases, I learned by listening to Matt Caberline's podcast about longevity and other topics that there were some cool experiments that were done in yeast and worms that showed that if you mess around with these tRNA synthetases, these Rs proteins, you can get effects on longevity, in particular, potentially lifespan extending effects. And these effects ostensibly don't seem to be attributed to, other metabolic pathways that we know affect lifespan, like nutrient signaling, which is something that rapamycin does. That's how it has its effects, or caloric restriction has, so it's not clear what the mechanism is of this tRNA synthetase longevity effect, at least in these simple organisms, but it suggests there could be this other avenue of metabolism that's worth pursuing that could provide additive benefit on top of things we already know that extend lifespan. So that got me thinking, well, it would be cruel if you could have these really simple yeast models of these tRNA synthetases, and lo and behold, we've been working on diseases where there are human beings. If they're missing these enzymes, these tRNA synthetase enzymes, they manifest a mitochondrial disease or another type of disease. So for 2 cases, FARS2 and NARS1, we've already run through this exercise and have worked with families and are working with families on translating the discoveries we've made, the molecules we've discovered. But then, of course, it got us thinking that if there is this real longevity hook, and it's interesting new biology, in fact, exciting, a new frontier maybe in longevity research that can be opened up, wouldn't it be great if we can at the same time help families in the process? And so yeah, I think there could be a really, really exciting opportunity to do the tried and true process we've worked out at Perlara to run these yeast avatar repurposing screens. We've applied them mostly to glycosylation disorders, mitochondrial disorders, but there's certainly every reason you could apply them to these tRNA synthetases. We've done it twice, in fact. But like I said, this alphabet in proteins at least has 18 other letters. So there's a lot of other white space to cover. And so that's the opportunity.
Yeah, that's a huge opportunity. First, the opportunity to help the families, and then potentially to discover a magical longevity drug that will make us immortal. Oh yeah.
Or, I mean, yeah, let's first extend lifespan in these kids under these dire circumstances, and maybe that's an avenue to extending lifespan in the rest of us. That would be the dream, of course, but we'll see if and how the science takes us there.
Absolutely. What I find especially inspiring about Perlara and Maggie's Pearl and all the spin-offs, that parents become co-founders in this venture. So it's a founder-led biotech, not a company led by venture capitalists, that allows to empower the patients, the people, to give them hope and to give them, I would say, some control over what happens to their lives. Do you think that this unique approach, I think I can call it unique, because practically you were a person who started to use actively in drug repurposing this N of one to N, I'm sorry, trials, do you think that one day it will become an industry standard for drug development or drug discovery for rare diseases?
Yeah, I think this is the start of a very long. Actually, I don't know how long it will take, but it's going to be a very comprehensive rebalancing of the healthcare ecosystem, and there will be much more control taken by individual families. We see it first with these early adopters, right? The early adopters of precision medicine are these families because they're so desperate. But if we believe everyone will eventually benefit, then there will be sort of the next wave and the next wave of adoption. So I think as business people see that running baby biotechs is actually profitable, and when you do this with many parallel opportunities, that this actually is a really interesting ecosystem. Once pure business people just recognize that will increase the adoption rate. Because once money senses that this, once investors sense there's money to be made here for real, not just hype or vaporware or hopium, but you can show them P&L statements and show them multiple examples, predictability of this type of business, and that will certainly really expand us. And then in the process, we'll open up precision medicine to more people. I mean, you can already see that. This is at least my hot take now. That we see the beginning of this N of one one to N transition. We see it first with young people, with children suffering from rare diseases, but just the demographics are going to start to allow us to see a new wave of N of one open up on the other side of the spectrum, as people who are older, right? Like, for example, my dad, who's 71, he 72. He actually, he is internet. He's not internet native, of course, but he was an early adopter to the internet. So very tech savvy, someone who, could find the resources in today's world to do something about, his aging process. And of course, he's had to deal with medical issues and stuff that happen as you just get older. But you can imagine now there's going to be a whole bunch of baby boomers and people of that age who are tech savvy enough. And as Father Time ticks on and they start to battle things like dementia or other things, they're going to have a much lower risk tolerance than maybe even people just a little bit older than them would have been. And you could start to see demand for N of ones to start to increase on the other side of the demographic spectrum, as people who want to not die and stave off, Alzheimer's or Parkinson's might be willing to try more and more radical treatments because they know that what the alternative is. So I've already, the first place I've seen evidence of this is with ALS, which obviously is usually an adult onset disease. But you can already start to see that's a preview of just people who will start to suffer from common neurological disease. They will start to take N of one risk, and that will start to be more normalized. So I think it's inevitable. And then I think Gen Z is so digital native and so internet native that they will just start. Some of them are just are tracking themselves, have been tracking themselves maybe since they were teenagers. So you have a whole group of people demographically coming up who are going to be like little mini Brian Johnsons, and that will just be the table stakes for people going forward. That they're going to kind of do some version of what he does in terms of tracking. So N of one to N. There's no turning the tide back. This is part of a massive realignment that's going to happen. But again, the leading edge of it has been these rare disease parent founders. And you're right, they are exactly another type of founder-led biotech that right now doesn't really get talked about, because we talk about the postdocs, we talk about the professors, we talk about the mid-level managers from Pfizer who go off on their own. We talk about all of them and we see them as founders, but I'm like, hey, people, there's this whole group of founders that have all the attributes. You just don't quite see them because you see them first as parents. But once you see through that first layer, you realize you're dealing with a peer.
Yeah, it looks like a Cambrian explosion awaits us soon. So, Ethan,
I
how
hope so.
does it feel to be a pioneer? of all of that?
Well, again, uh, I want to see results, and I want to stand on my accomplishments. So I know much more work is yet to be done, and I look forward to sharing those accomplishments and taking a victory lap some point in the future. But right now, there's still way too much work to do, and proof of concept is very great and very validating. I don't want to diminish everything that we've done. I've almost gotten a little desensitized to it, maybe didn't been too close for too long, but I also just want to stay focused on doing more and doing it faster.
Yeah, and we all will keep an eye on what Perlara and you are doing. And of course, also helping as much as we can to save the kids, yeah, and finally cure the rare diseases. Thank you so much for this talk. It was very insightful and inspiring.
Thanks, Maria.